Okay, everybody, it is February 3rd, 2007, about 5 o'clock Pacific time, and Roohif here wants to explain some of the experiments he's going to be doing, trying to sequence the pig genome, looking for the telomeric repeat that he believes there, or excuse me, the telomeric fusion that he knows is there, and to kind of debunk some more of Dr. Tompkins' work. Also, we're going to talk a little bit more about C. Brown and his that he's throwing out there and assertions that he's making without any scientific evidence whatsoever. Matter of fact, actually no evidence of any kind whatsoever. He's basically reached the point where he's just saying Dr. Schweitzer's wrong, and that's it. He doesn't explain why. He doesn't explain any of the science. He says, oh, your experiments are invalid. Well, that's not how it works. I think he's trying to solidify his position for Dumb Fuck of the Year. He's doing a damn good job. I'm going to make sure that he's nominated this year. I don't know if he's going to win, but I'm sure the health going to make sure that he's in the running, because I mean, he is literally, him and a few other people recently have come out of the gate strong of saying some of the most absolutely dumbest shit imaginable. So anyways, we're going to talk about that, but let's get started with Roohif here. Roohif, what is you going to be doing with this minion thing that you're getting and all this other chemicals and ethanol and shit you're buying? What's your experiment? What are you trying to show here?
What's the goal? Okay. Can you screen share me for a second?
I can do that. You are screen shared.
Thank you. So this is a article that Jeff Tompkins put out, I think a couple of years ago now, about the chromosome fusion, and he put this out in 2013. In it, he says that this is talking about the human chromosome 2 fusion. The alleged fusion sequence contains a different signature, a telomere-to-telomere fusion, and if real, would be the first documented case ever seen in nature. Obviously, in our chats over the last few months, people have listed various other examples from the literature that are clearly telomere-telomere fusions. I've found one in the pig, in the domestic pig, so Suscrofa. Am I still being screen shared?
Yeah, you're still being screen shared.
Cool. So this is a paper from 1996. So recent fusion events during evolution of pig chromosomes 3 and 6, identified by comparison with the Babirusa karyotype. And the Babirusa is like a wild pig from Indonesia. And where do we go?
So it's screen share, but it's still showing the ICR article. Is that intended?
Oh, okay. No, I'm trying to show the paper. 1 second.
Okay, yeah, you're not showing the paper. You're showing the ICR article. And by the way, people, in case you're wondering, I'm using the old method just for this particular Hangout, one, because it's faster to get started. Two, it's much less intensive on my computer. It doesn't like over process it, so I don't have these dropouts like I've been having, and I was able to invite more people. So if you're wondering why I'm using the old format, that's just why.
Yay for the old format.
Yeah, every so often I'll use it. I mean, like I said, it's so much easier setting up this type of format than the new one, and also, like I said, my computer can't handle it, so this one it has no problem with. Okay, so what are we looking at here?
Okay, so this is a paper from 1996 where they compared the karyotype of the domestic pig with a Babirusa, which is like a wild pig from Indonesia. So this little section here, oops, all right, just around here. Likewise, we conclude that pig chromosome 6 was most likely derived by a telomere-telomere fusion of ancestral chromosomes homologous to Babirusa chromosomes 6 and 14. And the evidence they give for that is a bit further down, or they talk about it for a while, but I mean, the visual evidence and here we go, this one here. Oops. So it's showing pig chromosome 6 is this one. And then in the Babirusa, chromosome 14 and chromosome 6. And you'll see there's little markers here. So one is called BHT164. These are little DNA markers that come up in fish, so fluorescent in situ hybridization studies. So BHT164 shows up near the telomere of Babirusa 14 and also near this little section here. Right next to that is this BHT 10.3, which shows up near the telomeres of chromosome 6 in the Babirusa, but also right next to the other marker in the pig. So it looks fairly obvious that this is a fusion site.
So when they look at the fish, and they'll see that the end there, the BHT161 is lit up, it's fluorescent, and then the other one, the BHT103, and the individual chromosome 14 and 6, and then they look at the pig chromosome 6 and find the exact same thing
right next
back to back as a telomeric fusion. Okay.
Yeah, so those markers aren't necessarily TTA, GGG, but they are near the telomeres. So, they're near the telomeres.
Genetic markers, whatever the genetic marker is for it. Okay.
Yeah.
Mm-hmm.
So they're near the telomeres in the Babirusa, but they're right next to each other in the pig chromosome. So I went looking in the current pig genome to try and find the actual fusion site, and the problem is that the pig genome is fairly incomplete. So it was made up of a whole bunch of bacterial artificial chromosomes, like hundreds of thousands of them, but not many of them overlap. So what the pig genome looks like is a whole bunch of these, spurts of 150 kilobytes or so of base pairs, and then they insert a gap between each bacterial artificial chromosome. So it's a bunch of backs separated by gaps of an arbitrary amount of 50,000. The fusion site is in one of those gaps based on these markers.
Rational mind doesn't like calling them gaps, by the way. I'm sorry. CRISPR doesn't like calling them gaps. The artificial gaps for alignment issues. Is that what you're referring to?
Yeah. What would he prefer they called?
Yeah. I don't know. He just said that he wouldn't call them gaps.
Okay. They're artificially inserted.
He says they're not gaps because we have sequenced that, and therefore they're not gaps of information. They're just artificially induced there for alignment. He doesn't like the word gaps, but I don't know one way or another. I don't care.
We don't have the sequence for those spaces. We don't
these
have,
right here. We do not. These are not been sequenced.
No, we don't have the sequence that closes those gaps.
So the fusion site, the fusion site on pig chromosome number 6 has not been sequenced then.
Yeah, there's no DNA in the current pig genome there.
Okay, and that's what you're going to be trying to sequence.
Yes. Yeah.
Okay. Let's call it unfinished sections.
So
do you have the base pair range, by the way?
The same.
Oh, I do not. I'll have to look it up. It's about the 50 million mark, I think. I've got.
I'm going to write something up before you do this experiment. You're going to write this up, and maybe somebody like Isaiah can actually kind of like follow along and do it at the same time. Kind of look in the databases and see what he finds as well.
Yeah, that's fine. Yep. I'll explain how I found where it should be. Yeah, that's fine.
Okay.
Yeah, so I have bought a MinION sequencer, and I bought plenty of other lab equipment, which is on its way. There's basically three components four components to the experiment. The first one is getting an actual sample of pig blood, which I'm in contact with a few people trying to get that when it's needed. Then I need to actually extract the DNA out of the blood, and there are blood kits made by various scientific suppliers. I bought one from a company called QIAGEN, and basically what it does is you put the blood into a little PCR tube, or a little 2 ml tube. Then there are some chemicals. One is called proteinase K, and basically that breaks down all the cell walls and stuff and releases the DNA. Then there are some magnetic beads which actually bind to DNA, and then you mix all that up and put your tubes into a magnetic rack, and obviously the magnets attract the magnetic beads with the DNA in them, and that enables you to get rid of all the rest of the crap, like all the proteins and the lipids and whatever else is not DNA. Then you basically wash it and get rid of unlock the beads, as it were, and then extract the DNA into another tube. So you've got purified DNA in solution. That's basically step one. Step two is getting it ready for minion sequencing, and the way the sequencer works is there are tiny little molecular machinery components called nanopores in the sequencer, and they basically pull a strand of DNA through the nanopore and keep track of the electrical impulses as they go through. So every nucleotide that goes through this nanopore has a slightly different electrical charge, and as it goes through, the sequencer records those charges and then attempts to do a base call on them. But to get the strand of DNA to go through the sequencer, you need to attach an adapter, and that's basically step two of the whole process, is using the MinION sequencing kit to place adapters on the end of each strand of DNA so that they can find their way into the sequencer, into the nanopore. So that's basically step two. Step three is actually doing the sequencing. So it goes through the nanopore, and then the computers basically work out or attempt to make base calls based on the electrical sequences, and it gives you your DNA sequence with corresponding quality data. And from that point, you're purely on the computer, and from there I'll attempt to actually place those reads onto the pig genome, and then hopefully find a fusion site.
Now, when you do this, are you going to be sending this to Tompkins? Because, I mean, you already showed with the Munchak deer that obviously fusions do occur. I mean, he actually said, as you pointed out in the ICR article, he actually wrote that these mythical telomeric fusions don't happen, right?
That's right, yeah. The Munchak is a telomere to satellite DNA fusions, and 6 of them, 6 or 7, whatever. So it would be nice to see an actual telomere to telomere fusion and what it looks like. I'm hoping it's very similar to human chromosome 2. There has been a study on the pig to show that there is actually interstitial telomeric sequences at the fusion site in chromosome 6 of the pig. So I know there's something there, but whether it's only one-sided or whether it's two-sided, I don't know yet. And I hope it's two-sided. But in any case, it'll be nice to actually flesh out the pig genome and cover some of these gaps.
Well, this won't change anything as far as the people in this community that don't understand that human chromosome 2 is a telomere fusion, right? I mean, they've already shown the actual sequencing of that particular, it's like 13.1 or something.
That actually shows or something.
Yeah, so I mean, we've actually shown them that obviously it's head-to-head fusion. They don't accept that. Now, this isn't going to change their thing, but if you can conclusively show that there was a telomeric fusion pigs too, are you just trying to show Tompkins that is not thing that is extremely impossible, that it has happened, especially in mammals?
It's definitely happened. I mean, this one's quite clear that it was a telomere fusion. Like, you can see the other centromeres in the two chromosomes of the Babirusa that are separate. So it's quite clear that it's a telomere fusion, but whether it left the same signature is the question.
Now, I was going to say, if I can ask a question on this, would you be willing to also go to the second step, is if you can find the precursor or related precursor species, or would you be willing to go look for the one where it was where the two things were separate, and then you can show that if the sequence lines up, if the two separate secrets lines up with the fused one, and I think that would go even a lot farther to say, hey, look, we suspect this to be a cousin species. It has a very similar matching genome, at least in that section. And it matches up as two separate parts, but when you go to the pig, they're together.
Yeah, like I'll definitely have a look for the second centromere. I actually haven't looked at it at all yet, so I don't even know if it's there or not. But I'll definitely do that. I mean, I think the pig and the babirusa can interbreed. So there'd be no doubt pig babirusa hybrid.
Yeah, because that'd go a long way to cementing that. Because
well,
I mean,
it would show that
you're
they're
gonna
definitely. It'd show they're definitely related and of the same biblical kind, have a common ancestor. So yes, there are babirusa pig hybrids.
Cool.
Yeah.
Yeah, I just thought I'd bring that up because there might be the odd person that would say, well, how can you not just think that that might just have occurred by chance? And it's like, well, if there's another species, yeah, that's why I posed that question, because that would go a long way.
Yeah. Yeah, so they're separate in the babirusa and fused in the pig, and if they're the same biblical kind, then it's clearly a fusion.
Is that the reason why you think Tompkins doesn't at all want to ever acknowledge that these fusions exist, because it somehow affects barremonology and what they consider to be a kind?
That's where we're getting into, is the fusion evidence of common descent, and I'm leaning. I'm sure Rational will be happy to hear that I'm leaning towards yes. I'm not firm on it, but I mean, if it is evidence of common descent, then yeah, I can understand his. I can understand Tompkins.
Trepidation.
I'm trying to think of a nice trepidation. I'm trying to think of a nice word.
He would have
his
to go down
hesitancy
that route, because if you show, I was going to say, if you show the link between the two related species, then you can do the next step. You can, if he keeps denying it, you can go from that related species to one that's a little bit more distal. If you're still finding the genome, and the sequences are similar, have very few changes in them, you can just keep going, and he's either going to have to make all animals just one kind, or he's going to have to say, okay, you might have a point.
Yeah, it'll be interesting, like you said, if I actually sequence the babirusa entirely and get an idea of what the substitution rate is between the two. So if humans and chimpanzees have a substitution rate of about 1.2%, if the babirusa and pig are greater than that 1.2%, they are definitely related, but then he would lose one of his legs to stand on about whether humans and chimpanzees are related.
Which he doesn't have any ground to stand on that anymore. I think that was pretty well refuted.
I mean, it's true, but I mean, the only sort of empirical way to show that they are related is if they can breed together, and that runs into all sorts of ethical problems. So we can't actually demonstrate whether humans and chimps can interbreed.
Oh, that's something that Tommy Hall would probably want to experiment on. But
so I'm doing the pig first.
We already know humans could breed with pigs. Haven't you ever seen something?
Oh, I've heard that one. Yeah.
Somebody had to ask, when do you think this will come to fruition? When do you think you have definitive experimentation results?
Like I said, I've bought everything. I actually bought the MinION.
Within a month?
Say again?
Within a month?
Maybe. It kind of depends on how easy it is to get the ethanol license and a pig sample. I bought the MinION back in December, and it still hasn't arrived. Everything else should be here within about a week or two. I mean, a month is possible, but it might be pushing it.
Yeah, well, okay, 2 months.
I would suggest 6 months. Well, you should come back at this in 6 months, because then that would be better. That you can double check on anything he has any concerns with.
Yeah, I'm happy to announce when I actually have sequence out of the MinION, and then go from there. But I mean, it'll take me, after I actually get sequenced, it'll take me a while to first of all, check for human DNA contamination, writing the software to actually join the two genomes together. Join my reads together with the current pig genome, and validate that. So there's probably a month's work of computer work putting it all together. But I mean, getting sequence could happen in a month, but it's most likely going to be 2. I was speaking to Steve before. I think I actually need a license from the Australian tax office here in order to get ethanol for the experiment, which is an odd place to get it. But the tax office here is wary of people buying ethanol to then make alcoholic beverages and sell them without the tax on them. So I should have a license to buy some ethanol, like only about 1 litre, in the next few weeks, I hope. And then I'll get a pig sample, and then hopefully we'll go.
Nate's, who's a PhD student actually in biology, actually recommended that you order primers surrounding the region and send it off somewhere for deoxy sequencing to confirm the results.
Say again?
He said you should actually order primers surrounding this region and send your sequencing off to confirm your results.
Yeah, I could do that, definitely. I'm an absolute newbie when it comes to going into a lab. Like I've never set foot in a lab. But yes, I could get a primer. So I could get a primer. Say again?
This is the first wet lab you've ever done? Wet lab work?
Yes, yeah. Completely. So there's a bit of experimentation just to get it going. I could get primers. I'm not entirely sure what primers I would get, purely because there is a massive gap in the sequence. So I mean, I could get a primer from the end of one of the back sequences, and just hope that it covers the fusion site. I'm not familiar with how to do that yet, so I'll have to look into that. But first of all, I just want to make the pig genome more complete, more so than I want to find the fusion site. I think the fusion site will be a nice artifact that comes out of just getting a lot of sequence. So I think that will work. If it needs confirmation, then sure, I'll try and get a primer and extract it and PCR it. I think. Go again. Yeah.
Oh, no, go ahead.
Yeah, I was the thermal cycler that I bought. I am hoping can do PCR as well. I know that it can be programmed in a basic sort of manner, but whether it can do full PCR, I'm not sure. I think it can.
Some people in the market are actually asking if you're a creationist or not. I'm like, no, Roohif is not a creationist. No. If you guys have not known Roohif by now, something is seriously wrong. You don't watch my channel enough. No, Roohif is definitely not a creationist. He actually is the person who basically pointed out to Dr. Tompkins that his genomic analysis between chimpanzees and humans was utterly flawed. Not so much because Tompkins made a mistake or anything, but because the program in BLAST had an error.
Oh, he made a mistake.
He made a mistake. I'm trying to be charitable. He did. No. He did admit there was a program. The problem when there's a BLAST with that, when the batch programming, but he made mistakes subsequently to that. He's acknowledging that. Although I don't think he fully gave you credit as the person who pointed that out to him. Did he?
And if I can make a statement here, I would say to that person who was asking if he's Christian, even if he was doing the work properly, he was being honest about it, it wouldn't matter whether he was a creationist or not, as long as he's willing to follow where the evidence was. So, calling somebody a creationist is just moot, or questioning whether somebody is or not. If they're doing the work honestly, and they're saying, well, whatever the results is, I'll go with it, and they do that, then that's all. Then it doesn't matter what position they are. At least that's my thought.
Yeah, I do think that Tompkins is dishonest in his work.
I was saying the same thing to, I think it was CRISPR the other day, and again, I used to be a lot more charitable than I am now. I think this year I'm actually losing a lot of my charitability, because after talking to him and actually reading that email correspondence between him, you, myself, and Dr. Anderson, I've really lost the will to even give these people any kind of charity. I just find them to be very negative, and definitely you see a huge difference between the people we've dealt with people like the Reasons to Believe crowd, the older creationists, and these younger creationists with ICR and AIG. It's a completely different feeling, right? I mean, when I'm talking to like Anastasia, which is the secretary for Reasons to Believe, very nice, very pleasant, very helpful, from ICR or IG, and I have called them and talked to them. I don't get that at all. I don't see the same response as people saying, hey, you know what, we really want to get some firsthand information. Can you provide us a person that can explain some of the things that you guys have been saying? They're not willing to do so. Corporations will not put themselves out there with experts on some of the technical stuff they've blogged about. I've never seen it. Have you ever seen it anywhere on YouTube of Dr. Tompkins or any of them really talking about the technical specificities of things that they blogged about with anybody who might be able to give them a run? No.
They want one-way conversations, and that's sad, and it's telling.
It is sad.
And telling.
I'm just going through the live chat on the outside. Scientist Sam, is that Roohif? Yes. I am a shadow of my former self, though, at the moment. Natase is asking me to get a primer, basically, to try and sequence the region and do a Sanger sequencing. I could, but as you say, like, I would need to be within about 700 base pairs of the location, which I'm probably not. The gaps, the artificial gaps between these backs in the genome are 50K, and they could be, could be several hundred K. If I could create some backs with a primer at the end of one of these pre-existing backs, then maybe I could cover it, because if I can get a back to last for 160K, then I'll probably cover it. But I have no idea how to do that yet, so I may have to do some research if this experiment doesn't yield what I want. Leighton Bacon, Roohif is poorly. Yes and no. I'm getting better. I've just come out of a stem cell transplant, so I've got no hair, and just very weak and tired.
And he still spends time with them.
Yes, yeah. Roohif looks like he has a quadcopter. Yes, I have one of those. I also have one of those.
Some badass tours, doesn't he?
Oh yeah, good. I'm jelly. I'm very jelly. Hey, he's doing lab work. I'm extremely jelly here.
Where are we?
Computer, something from like Star Wars. It's like futuristic. Do you still have that blade server thing?
Oh, I've got 4 of them. Yeah.
Yeah, I mean, he's got freaking blade server.
Yeah, they were for.
It's like you have internet backbone in there somewhere.
No, I'm actually running over ADSL. Sorry, this is getting a bit off topic. My career job is as a high frequency trader. So I've written software to trade derivatives on the Korean market. So it's latency sensitive at one end, but not latency sensitive between Sydney and Korea. So I've written a protocol specifically to be fairly tight over the wire. But that's
are you still doing that? I thought you kind of like.
Well, I was looking into it late last year, and I was getting close to trading, and then I got diagnosed with cancer, so it didn't happen. I will probably take it up again at the end of the year when I'm feeling better.
Oh dear.
You can make a homemade PCR if you need to. Just water baths at the right temperature.
No, he meant a thermal cycle.
Yeah, I've bought a thermal cycler from Eppendorf, which I believe can do proper PCR cycles. I know that you can program it in sequentially. So it's, it may just need a little more manual intervention than a proper PCR cycler. I'm not sure. I am most certainly not a young Earth creationist. Certainly not.
You guys don't watch Steve enough.
Has Roohif received any technical questions
FYI. Nor have I ever been a young Earth creationist.
I'm jealous of that.
Do I have a channel?
Well, Max,
Hang
you
on.
used to be, and I think Opal, weren't you at one time too?
Well, kind of. The whole Mormon thingy.
That's not a young Earth.
Yeah.
Yeah, it is. Don't Mormons? I know a guy whose mother was
Kind of.
Mormon, and she thought the Earth was 3,000 years old.
I've never been any Mormon that thinks that, and I've known hundreds of Mormons.
Well, the ward we were in, they never really went into that at all. They kind of left it vague.
Oops.
They just, kind of, accepted, Genesis and everything. And
Well, you might be right on that. I mean, there are some people, regardless of their religion, might actually have some kind of latent thoughts that the Earth might be within, 6, 7, 8,000, 9,000, 10,000 years old. And I just recently kind of thought that because I play WoW occasionally now. And one of the people I play with is an old roommate of mine that actually used to sublet from me. And he is becoming an electrical engineer. He's like in his 4th year of differential equations and mathematics. Smart kid. And he was very religious, always been really, really religious, really Christian. And, we used to talk all the time. And I never asked him really about the age of the Earth. It really never came up until yesterday. And I was like, by the way, I've never really asked you. I mean, what do you think the age of the Earth is? And he's like, well, I don't really think science says it's as old as they say it is, but I don't think it's 7 to 10. I'm like, well, what does that mean? He's like, well, I know it's probably just a few millions or something like that. I'm like, wow, that's like where do you even get that from? It's like to believe a young Earth creation that is the Earth is 7 to 10,000. Thing out of your ass from like, oh, I think it's just like 200 million. Well, based upon what?
Well, because it kind of feels right. Like, the scientists can't be right about what they're saying because it goes against what I might think, but what I might think sounds stupid. So, 200 million.
Well, that's what C. Brown does, right? He has one big argument for incredulity and emotion, saying, "Oh, well, collagen can't last 65 million years." Okay, why? Can you explain to us?
Says why?
Yeah, I mean that's what I've been asking him. I've been asking him consistently. Can you explain to us why collagen would break down? There's no collagenases. There's no bacterial component to it. There's nothing exposing to the environment. It's basically hermetically sealed and in an anaerobic environment. What's going to cause collagen to break down? Roohif, what would you think collagen would magically break down? What those peptide bonds are just going to fall apart? Because why?
You're asking me that?
I think it was a rhetorical question.
It was rhetorical, but I mean, why do you think Roohif thinks that? Why do you think that C. Brown thinks that the collagen is going to magically break down?
I have no idea. I seriously know fuck all about chemistry.
The placebo effect doesn't affect chemistry around you.
Okay.
Placebo effect. Well, I'll ask Urology. Urology, how about you? What do you think? Do you have an idea why C. Brown would magically think that the peptide bonds, the amino acids of the protein would just spontaneously just break apart and cleave?
I'm not too sure about chemistry myself, Steve. So, I only know the basics, unfortunately.
Well, anybody wants to answer that in the live feed. Maybe Natase has a PhD in biochemistry, so maybe he might want to throw that around. Because I don't know why C. Brown thinks it's going to break apart, right? I don't know. I mean, he's never explained why he thinks collagen is.
C. Brown always knows a lot of things, except all of the things that he knows is just full of shit. It's just shit.
Correct.
Well, Steve, we can't know that the laws of physics were the same back then,
I've had similar arguments, or that the laws of physics aren't the same in other parts of the universe. My favorite
which means fuck all, though. Which totally means fuck all, because if you don't know what it was back then, how the hell you make an adjustment for what it is now, or what they're trying to impose?
Precisely.
Matthew Ecclesiastes said that how do we know that 2 + 2 or 1 + 1 equals the same here as it does in other galaxies? And I'm like, are you really positing that?
If you look out through a telescope and you see one galaxy and you see another galaxy, how many galaxies are there?
That's a good point. I should have brought that up to him. Thanks. Better late than never. No, he doesn't understand that it's just an analytic proposition. We define it to be that way. Therefore, it doesn't matter where you are in the universe. By definition, 1 + 1 = 2. So, off topic. Roof's like, I got up for this.
I got to answer the rest of these questions in the live chat.
By the way, answer to my question. If he answered, I don't know, I give you the right to smack him.
Which question?
What?
When you see two
I think they say
galaxies, you see one galaxy,
Oh.
then you see another galaxy. How many galaxies are there?
Yeah.
Now, one guy in the live feed, Haroon, says, I've seen an argument from a young Earth creationist that the second law of thermodynamics guarantees college to break down even conditions. Why?
What?
Thermodynamics apply to college breaking down. I don't see how they keep throwing these things around like the second law of thermodynamics has anything to do with that.
College, yeah.
I'll tell you what the second law of
colleges-
thermodynamics has to do with. Thermodynamics.
Yeah, I don't know.
One's a biochem and one is about physics. Yeah, good luck breaking that one down.
Yeah, I mean, they don't seem to understand that the second law of thermodynamics only really applies to isolated systems, and the only isolated system in the universe. So basically, all the second law is saying is that no matter what happens, the net entropy to the universe must increase.
Okay,
Well,
which reminds me.
You're missing the whole thing. That argument does prove a breakdown of their argument.
Beta decay in collagen.
Oh, okay. Also, I sent you a picture from that somebody would posted, like some kind of essay about entropy or argument against determinism, as they put it. And I sent it with the caption or subtext that said, "They said entropy. They used entropy wrong."
I'll look at it. What were you saying from the live feed?
I was just going to finish answering some of the questions. Where are we?
Go ahead.
So Trevor asked, have I received any technical questions about his experimental procedures from C. Brown, which will inform the investigation? No, I have not. Haroon said, or Said, if Steve, could you ask on my behalf on how one goes about being an independent scientist?
You don't need no man.
I don't know. I don't know, and I'm having a crack. So, I mean, I hope to write a paper on this, and we'll have an attempt at submitting it somewhere. I have no idea how that will go.
First order of business to being an independent scientist is to be damn well humble.
Yeah, I'll pay that.
And skeptical of your own findings too. That's one of the things that I liked about Mary Schweitzer. She was very skeptical about her own work.
Yeah, so I don't see why people can't be independent scientists. I mean,
Flat earthers.
Well, submit it to peer review, see how it goes. Get smashed.
And you don't have to be a scientist to be scientific, right? I'm far from a scientist, but I like to consider myself scientific in thought, right? So I think that most people can be scientific and just apply scientific methodology, just apply critical thinking. Because if you don't know what it is, then you are a member of the clown car posse by now. I mean, we all kind of explain to people what it takes to have a critical mindset. And went around to, like, you just recently messaged me, just literally like a minute ago, said, you know, from what we know about decay, there's no way these things could last. Well, what do we know about decay that states this? He's not using any kind of scientific methodology or even rationalization. It's just argument from incredulity. He's just saying they can't last. Well, why not? Something has to break something down. There needs to be energy put into a system to break something down.
Steve, because they can't.
But that's not a scientific explanation, right? I mean, things like,
but it makes
there
me
has
feel
to be a
good.
reason for something, and they just can't say, okay, well, if there's microbes, sure. If there's collagen, sure. If there's something enzymatic happening to break this stuff down, sure. But if not, why? That's like saying I have a block of iron 50. Well, okay, that can't last forever. Well, the end of the universe, right? I mean, obviously the universe is going to eventually, you know, be a sea of photons, but why can't a block of iron last billions and billions and billions and billions of years? There's no reason why. Stuff can't last this long under these certain conditions, but won't explain why. It's no different than me saying, okay, a block of iron can't last billions of years. Well, why can't it? Now, other things can't because of decay, but iron 50 is stable, so that's why. You know,
neutronium is even more stable.
Neutronium. Man, can you imagine if we actually were able to experiment with this microscopic piece of neutronium? I mean, it'd have to be like really still weigh a hell of a lot, but
yeah.
Well, you know, we might be able to produce neutronium, but we actually supposedly are working towards producing metallic hydrogen. Now, I don't know if they've actually successfully be able to produce a metastable form of it, but they're talking about but I don't.
When did they actually do this?
I
2 weeks ago or so.
I read a
2
couple
weeks ago.
articles. They said that
Yeah.
they did create it, and it's something that never existed on Earth before then.
I don't agree with that though. I've looked into that after I saw the news report, and they have not confirmed that's what it is. They think
I'm pretty sure there was other attempts. There was previously other attempts, and
they failed.
Yeah, none of them actually. Yeah, none of them worked.
Yes, but this is something that's out there that they really do think that they might have produced it, but they have not been able to confirm it. And so any news report saying that they produced this is wrong. And that's what happens with pop science, because they'll come back and do a retraction and say, "Oh, look, we made a mistake. They didn't produce this." That's why scientists don't say things like that. You don't find scientists saying we've discovered this or confirmed it until it's been confirmed. That's the difference between pop science and actual science.
Well, has anyone confirmed the time crystals yet?
The what?
The matter that persists in time.
No idea what you're talking about.
Oh.
Well,
Somebody says my audio is having some compression issues. I'm sorry, my computer's a piece of shit, Tyler. What do you want? And it still is probably better than it was, because when I'm using the other format, just completely, I can't even like do a Google search because my computer just bogs down to nothing and it drops the feed. But anyways, with the metallic hydrogen, excuse me, urology, yeah, look into it for me, because like I said, I've looked into it a little bit. I don't see anywhere where they've done a confirmation of this yet. They said it basically. They've said it looks like. Well, this is the same thing we're running in with the red blood cells with Dr. Schweitzer. It may look like red blood cells, but you cannot say it until it's verified. And there's been reasons why they haven't been able to verify that they're red blood cells yet, and I remember why. Now, eventually, will they? Maybe yes. If they are, they confirm it. That doesn't demonstrate a young Earth. It doesn't change anything we think about the age of the Earth. But since Steve Brown it does. Oh well, if they find red blood cells, that means that evolution is wrong. No, it has no effect on it whatsoever. But they put a much higher evidence on it that if the red blood cells come back as actually being red blood cells, that the age of the Earth must be wrong. And I have no idea where they get that from. It doesn't change anything. But it still has to be validated that they are red blood cells. It's step by step. So my argument to them is that you cannot even say it's red blood cells yet until it's confirmed. That's the difference between a scientist and pop science.
Also, not only that, if you want to even say, you got to then show that that any condition that is found that they were found in will not preserve them, because there are mechanisms to preserve stuff. Sometimes extreme low oxygen. I'm trying to remember, because it's been a while since I looked in it, but there's a lot.
Well, there's like 7 or 8 things that preserve this particular samples.
Yeah.
A lack of oxygen was one of them. Lack of bacteria, formaldehyde, and a few other things.
And not.
But you see what I'm saying? When you read a pop science things that says, "Oh, they found red blood cells," or they found metallic hydrogen. Well, until a laboratory. That's a headline, right? That's why you can't go by that stuff. Don't do it.
Yeah.
Matter of fact, you guys remember when they said, "Oh, the speed of light has been violated"? Scientists discover faster than light travel, and it ended up being like a 60 nanosecond difference, and that was finally contributed to a loose cable in the sensing mechanisms.
Yeah, it wasn't faster. Nope.
No, but they had actually news reports saying scientists discover neutrinos can go faster than the speed of light, and I'm like, no, that's bullshit.
Yeah, I even remember when they came out, they were just putting it out there for somebody to help them figure out what was going wrong. They're just like,
right, a year later after the fact, which was stupid, but yes. But pop science ran with that, saying, "Oh, look, it's violating the speed of light." Scientists, found neutrinos go faster than the speed of light, and when you actually look at the article, that's not what the scientists were saying at all. So,
no, but but like most news media this day, they just need the clicks. They need the people to come in, and the sad thing is most people don't read beyond the first paragraph.
Sadly.
And they just grab the headline and just start running with it. They start inventing the story in their mind, and I'm seeing that way too much.
So, Roohif, what do you think Dr. Thompson's going to go new quote unquote peer review paper is going to be about?
I heard it's about me, so I don't know.
Yeah, well, yeah, he said it's about, well, he said it's in response to what you say. I don't know if he's actually going to address.
I'm just curious on who's going to review it.
Well, his
someone at
ICR.
Yeah.
Oh, so it's not really scientifically peered reviews. It's just ICR peer.
Basically, yeah. I'm curious.
Like it's Sean peer type. Remember Sean says
like he's obviously said a lot of times that I'm out there talking smack and I've got no idea what I'm talking about and I'm wrong and everything.
Of course, you won't have anything to back that up, I guess.
Yeah, my main concern is that, like I said, he wants a one-way conversation, and my anticipation is that his paper will just be crap, and then I'll have to spend some more time showing why it's crap rather than having an actual discussion. That's how
creationists thrive. One-way conversations. When they have two-way conversations, they fail in court.
Yeah, I've already asked him to retract his most recent paper about the chimpanzee trace reads, and he knows why it's crap now. So yeah.
Well, when you say paper,
I don't expect him to.
Do you mean a blog or?
No. He actually published late last year, where he tested 101 trace archives. Say again?
He does. He has actually published papers, but the one he did on the human chromosome number 2 fusion rebuttal was never a published paper.
No, and it's a similar idea with this chimpanzee trace reads one, where he talks about the possibility of human DNA contamination in the chimp genome.
Where did he get it published? The paper.
I got it published in Answers Research Journal, which is Answers in Genesis.
Yeah, that's not
real
published.
Place.
Yeah, I know. So, I've already explained to him what the real problem is, and once I have, I then get no response from him.
It's basically like saying, I have my ad up in this Applebee's bulletin board, and the guy replies to you, I don't live near an Applebee's. That's like posting on creationist stuff.
Yeah.
Well, some of the stuff people publish, they expect to have people, criticize their publication, and true scientists should be able to defend it, their work. And younger creationists won't do that. Again, I have tried numerous times to get these people to come in and to defend themselves, and it would not be a fluff thing. It would be a hardcore talk about the technology or the technical aspects. Most of us would probably be very lost in the conversation, but it would be out there for other people that are familiar with the science to review it and go, shit, Roohif's spot on, or Roohif, you don't know what the fuck you're talking about. But that's what I would like to see happen, and Tompkins just will not do it, even though he said he would at one time.
Oh, and I imagine most of his stuff against me will either not answer my criticisms correctly, or it'll just be an ad hom attack. That's what I'm guessing.
Yeah, well, you're bald.
I'm bald.
Well, we have to. What's the problem is now we have, as you said, we have to wait another month.
Well.
Yeah.
Can you see how ghosty it's coming back?
No, you look like Lex Luthor.
Yeah, it's pretty bad. It's all ghosty, grey, old man hair.
I don't know. I kind of like your look. You got this neat look about you. I don't know why.
He looks kind of badass, doesn't he?
He does.
His eyes are a little dark. He's got a little raccoon. But other than that, he does look like the Lex Luthor badass.
Yeah, he either needs a tuxedo.
Once you start putting on, he'll look like a badass man. Like, I'll fuck you up, boy. I'll kick your ass. You don't fuck.
Yeah, he'll hunt you down and kill you if you kidnap his stepbrother.
What I don't have, he'll be like,
a couple of questions.
What I do have is a unique set of trait or skills. Yeah, what's that?
I'll answer the rest of these questions and then I'll shut up. Sarah Rodriguez, do I have a channel? Yes, I do, but there are videos of my kids on it. There's no science videos on it or anything. There's my kids' science video on there, so check that out. My little boy, who was 7 at the time, doing a science experiment. I do have a blog, yes. Obeyed, yes, I do have very sunken eyes right now. Obeyed again.
That's me. That's me. You know that.
Yeah. working in a derivatives prop shop, yes. Not at the moment, obviously, but I'm a founder of a company called Tibra in Australia.
Oh wow, yes, I know that one.
Yeah,
right,
he's a little bit of a muckety muck in Australia. He's
So it's Tibra. Have you heard of the?
Rue's got name recognition in Australia.
He's a real name down there.
I live in the same city he does. Come on.
Oh, you do. So you've heard of the court case between Tibra and Optiva?
Well, I know both those companies. In fact, I was actually going on an interview with Optiva at one stage.
Oh wow, there you go. Anyway, so
that was settled years ago, though, wasn't it? Though.
Yeah, 2014 or 15 or something. Yeah, a couple of years ago.
Nobody lives in 2004 anymore.
Yeah.
Yeah, if you live in Australia, you probably know who Roohif is or have heard of him.
Oh, if you know the industry, yes.
Yeah.
It's a fairly niche industry, though.
In America, we all know you before you were cool.
Leighton Bacon, do I like video games? Not particularly.
You're weird.
Yeah, what is wrong with you? Really? There's no game that you would like, Roohif? Come on, seriously. No video game at all?
No,
I got to introduce him to Ark. Ark then. Ark's
How about
Come on. Ark
Yeah.
out of all games. I just got done with Uncharted 4, and that game was amazing.
Yeah.
Well, you like flight simulators, don't you, Roof? I mean, like not a full size one.
I like
but...
actual flight simulators, like a
I know
proper,
you do, but
Yeah. He likes
But
pretending to
yeah.
fly.
I have a fear of flying, and being in an actual flight simulator helps understand what can go wrong and what you do when something does go wrong, and how much redundancy there is, I guess. So that'd
be an awesome toy.
Oh yeah, nuts. I mean, the last time I was in one,
Expensive toy.
I lost the left engine and the landing gear wouldn't come down, and I was flying into Caltech, Hong Kong, and I did a pretty good job of landing that. So,
how did you land it if the landing gear didn't come down?
I had to
belly in.
basically belly up. So I had to come in at quite a steep angle after I was very close to touchdown, and put the front landing gear didn't come down. So I landed on both back wheels, and then sort of coasted at that sort of angle, and then gradually put the nose down. With one engine.
Pretty badass. I would hate to have to be the guy to clean that up.
Well, that's actually a pretty decent landing. I mean,
across the runway.
Yeah, you can walk away from that landing.
Tied in. Yeah. What the fuck, Trevor?
Have you ever simulated when you're doing a landing an ILS and you have a crosswind and it's just basically like you're trying to align it and you're just playing blow off course? That's fucking hard, man.
Yeah.
Because you got to ping your outer marker. You have these markers that when you're doing an ILS landing, these outer and inner markers, and these are what you try to focus into because you have this small narrow, like I guess you call it a window, but it's a very small region, and that's what you have to focus the actual nose on to try to get to the landing. And basically, these things land themselves. Really, I mean, if you can get right on that glide path, you don't even need to do anything. The plane's going to do it for itself if you want.
Sorry, being a Berry asked Roohif, are you part Aborigine? I get that a bit actually. I am fairly dark, but I'm literally first fleet extraction. I'm looking particularly dark in the eyes after I've lost a bit of weight through chemo and stuff. So hopefully that comes back, and hopefully hair comes back.
You look less like a guy that smells. You'd look less like a guy who sells potions.
Okay.
I thought you said smells.
No, sells, not smells.
There we go. That is the end of the live chat. There we go. I'm not big down under. I am in the industry that I was in.
That wasn't a dick joke.
I'm known as the founder of a company. So a derivatives trading company. Hey Wayne, how you going?
Well, I was going to say before you got into answering questions, I was going to say the good thing about coming here and talking about it is most of us have high experience with the arguments creationists will have, and I think we will probably be big benefit to you because we can pretty much prep any write-ups you do for any challenges a creationist might give up.
Yeah.
Yeah.
That'd be cool. I do plan on writing a paper and submitting it, but I mean, I'm not sure how I'll go getting it published, given that I'm not affiliated and not actually a scientist. But I mean, I will try. And
I'm sure that Sam or Fiona could probably help you in that regard of how that process would go about being done.
Yep.
Yeah, because I'm sure there's others that have, that have managed to do the work and managed to get to the level where they've actually produced work that is publishable. So go for it. I strongly encourage you to try for it anyways. As I said, I think this community will be a great resource because we'll be, we'll be able to fill in for creationists without actually, making you put up with the attitude that you might get from some individuals.
Yeah. I mean, as far as
have you read his blog?
No, I haven't. I barely have time for a lot of stuff.
You should read his blog. He's got a very well written blog.
Oh, I bet. From what I've heard of him, he sounds like he would. So, but I will check it out.
Cool. I'll put it in the live chat. Hold on.
Yeah, I mean, his blog is just concise. It's it's. I don't know if the average layperson would. I think the negative thing on his blog is that unless you do know a little bit of maybe about it, the stuff, it's not an easy read, but it's definitely well written. I mean, I think you try to make it easy read, but I think you overestimate the reader's ability to understand this stuff.
Maybe.
That's the good side of the Dunning-Kruger effect.
Well, Ruth's like a genius, right? So he just kind of naturally thinks that most people are, like not complete morons. Although,
I write for people that are familiar with the arguments.
Yeah.
I write mostly for Tompkins. I think he's probably number one viewer, hopefully. And anyone else that's familiar with it.
Isn't that like keep your friends close, but your enemies closer? Having a fucking creationist be your number one viewer.
Yeah.
Yep. Well, when you start feeling better, you should start doing videos like Paulogia does towards Eric Hovind.
Who?
Well, Paulogia, Paul. Paul does videos specifically refuting Eric Hovind. That's his little niche that he has. Occasionally, he might delve into Ken Ham, but it's mostly about Eric Hovind. And Eric has actually invited him to come debate him. They do know each other. Or corresponded.
No, something about Eric Hovind's face pisses me off.
Well, he's got that snake oil salesman look always.
Yeah, I know exactly. It's ridiculous.
Yeah, that's why I think
talking to Thunderfoot. I wanted to shoot when he was talking to Thunderfoot. He won. What?
I told it things like to R and Raw. We kind of need each other because it builds up drama kind of stuff. I mean, I don't know what exact words he said, but Aaron was saying that he's like, no, Eric, we don't need you, and just a lying, duplicitous snake oil person. That's it, and he's a nice guy as far as like. Him? Well, I mean, not so many words. I mean, Aaron, he's pretty vocal about his opinion. He doesn't hold Eric in high esteem by any stretch of the imagination.
No, of course not. But if he said that to him, I was just gonna say savage.
But I think Eric knows that deep down he's full of shit. He doesn't care. He's making him money.
Which is probably why it makes me so angry.
Yeah,
I've be honest and stop lying.
I kind of moved on from that. It just got too stressful for me, and I like coming into things like this where people are going to do stuff, even if it was something that was kind of done, him doing it, is kind of interesting because I get to see how all this stuff works and get to learn a little bit. That's why I'm here. I'm here to expand. I don't mind the creationist debate as long as it's expanding and stuff like that. But when it gets repetitive after a while, when it's the same thing over again, and it's just believe me because I say so type attitude, that starts to turn me off.
So, there's also when it gets repetitive, but you don't even know what the hell the guy's talking about.
Somebody in the live feed, actually Ben on his live feed, he asked me something in the live feed, and I'm just too tired to type. But Sybaris was talking about me a long time ago, and that there's a thread that got brought up again by Ludic Interface and a few other people. I don't know anything really about it. I don't know what the beef was with Sybaris. I don't know. I don't remember even what the issue was, except that he just wasn't understanding what I was trying to tell him about atheism. But yeah, I don't remember what he kept asking me. He basically was trying to get me answers, ask some weird questions, and I'd answered them the best I could, Benna, but he wasn't accepting my answers. And I'm like, look, I'm not trying to be evasive, but the way you're asking the question, I'm answering it the only way that I know how to answer. And unfortunately, some people, if they don't know what you're talking about, thinks that's a non-answer, and it's not always. Sometimes you're actually giving a very valid answer, but the person asking the question doesn't understand the answer well enough to recognize it as an actual answer. Does that make sense? Anyways, but that's the deal on that. And then Wayne is asking about the expansion wave of the universe. The universe, as far as we know, is still accelerating. They still hypothesize dark for the acceleration. Yes.
Yes.
I think just the volume. I'm trying to remember. I think it's just the percentages or the rate is a little bit different, or at least they suspect that.
Yeah, the rate might be different. The rate of the Hubble constant change, but I mean the Hubble constant might actually vary differently the further you get to the observable event horizon,
which changes depending on your position. Oh wait, you said observable event horizon. Never mind.
Yeah. Well, the constant is the Hubble's constant really is a constant from the Earth as we observe it.
Yeah.
But that value, the value of things accelerating, it does change. I should say it's still a constant because it's actually measured in reference to us. But
right,
the rate of expansion does change.
Right.
Back to this pig stuff. So, I mean, does anybody have any questions about what Roohif's going to be doing here? So a month or 2 months or whatever it is down the line where we do a recap on this, we can kind of look back and say, okay, well, Roohif said that we're going to see this, this, and this. What do we expect? We expect to see a telomeric, right? Not a telomeric to satellite, right? For Roohif?
Yeah, hopefully a telomere to telomere, so a head-to-head fusion, hopefully.
So we should see something very similar to the type of head-to-head fusion that's on human chromosome number 2 then.
That is the hope, yes.
How degraded that region actually is? Do you have any idea?
How degraded?
Yeah.
No idea. I know that they've found, they've done studies to show that there are TTA GGG repeats there. I just don't know whether they are one way or head to head.
So you don't know well, yeah, but if you're trying to show there's a fusion, wouldn't you have to see if they're both way, if they're two way?
That'd be better, definitely. I mean, there are other ways. If I could get a hold of Babirusa sample, I could show how close they are to the telomeres. Like it's possible that one side of the telomere may have broken off a little earlier or a little shorter. Yeah.
If I don't find one way, it'd still be evidence, right? Good evidence.
Yeah. But I mean, I would have to do more work to verify it, really.
Gotcha. And if you got a hold of a Babirusa, if I pronounced that right, and you saw, paralleling sequences from each section, that would be even more evidence. I mean, that'd be really hard to ignore at that point. I mean,
yeah, there's levels I can go to confirm it. The easiest way to confirm it is purely just to see the head-to-head fusion. But if that's not there, if it's not head-to-head, then I need to do some more sequencing.
Hey, quick question, which might be a little embarrassing, but the question I have is why? Because I might have been sleeping. Sorry, Steve.
Basically, Jeff Tompkins makes a claim that there are no documented telomere-to-telomere fusions.
Okay, I wasn't sleeping then. Yeah, what a dumbass.
Yeah, that's pretty much it. And it's fun to learn and fun to do. So.
And you might actually show something, because if you can show a correlation between the two things, you may. I'm trying to think. There's a way that you might even be able to calculate when the split was, because
possibly. Yeah.
What are they called? Those insertions by those viruses that just fail to be viruses.
Yeah, endogenous retroviruses.
Yeah, that's it.
Yeah, I need to do a bit of work on that to figure that out. But yeah, it can be done.
Oh well, then there you go. Now you got if it turns out you got another project.
Yeah,
you might get some fame then.
The second
wait.
project would not be a pig; it would be the jack jumper ant. It's one of the only animals that has one chromosome, or can have one chromosome, and it's most likely a telomere fusion as well. So it's also
do you
handy.
think did it have more chromosomes at one time and just have multiple chromosomes?
Well, there are colonies. There are colonies where they have. There are colonies in New South Wales where I live, where they have somewhere between 1, 2, and 3 chromosomes. So I'll obviously try and find a colony where predominantly they have one chromosome, and there's papers going back to I think about 1985 or thereabouts, showing why this should be a telomere fusion as well. Obviously, it hasn't been sequenced yet, but if I can get a hold of those, I'll try and sequence those as well.
So you said Dr.
Things about him. I mean, he's just gone full troll mode at this point. I mean, she has a PhD. He probably doesn't have a GED, and he's trying to convince people that Fiona's intimidated by him. It's like, are you out of your fucking mind? You're a dumbass, dude. So this is why I'm saying that. By the way, keep in mind this for the dumb fuck of the year. This is one of the stupidest things that I've ever heard. Him trying to say that Fiona's intimidated him. That should be in the running for dumb fuck of the year.
That should just be his headline, just a picture of him or his profile picture with quotations. Fiona is intimidated by me. C. Brown.
Yeah, I'm sure there might be a meme of that at some point.
Yeah.
But... Sorry.
Okay. So you said Dr. Tompkins said there were no documented cases of telomeric fusion.
Yeah, that's right.
Yeah, he said they're mythical.
Aren't mythical? Okay.
Yeah. Yeah, he doesn't
Then why
accept.
don't you just show him that?
What's
Like,
that?
why work on pigs when you can just show him the human chromosome number 2?
Because he doesn't accept that one either.
He rejects it. Yeah.
Okay, well then show him the pig and then show him the papers for that. Or you could just throw a Bible at his face and say that this is fictional.
But is he going to use the same arguments to say, "Oh, well, that's not a telomeric fusion either"? I mean, he just adamantly asserts there's no such thing as a telomeric to telomeric fusion, except in maybe somatic cells, right?
He won't recognize the human or the he won't recognize the human as one because he thinks the fusion site is too small and too degenerate, and that's why I showed him the Indian muntjac fusion sites, which are also short and degenerate.
But that's ridiculous. I mean, you would expect there to be degenerative.
Yes.
Expect that to happen because that's what's caused. Like that's for a telomeric fusion to occur. How are you going to have a telomeric fusion if they're pristine?
Exactly, and long.
Yeah. I don't know.
Exactly.
Again, those are the questions I really wish people could answer him in real time or ask him in real time because I don't know where he's coming from on that.
Yeah, he won't answer me.
So wait. If I'm understanding correctly, it's like saying the moon can't be a sphere because you see craters leaning away from us.
I'm not sure what the analogy is there.
No, I'm trying to say but he's trying to say why it can't be is exactly why it is.
Yeah. The signature of a fusion, he would say, must contain thousands of pristine repeats of the telomere sequence. When if there were actually thousands of repeats of telomere sequence, there would not be a fusion in the first place because the telomere repeats would have done their job and formed the telomere complex to stop fusion. So
he has a PhD in this. How could he be so blind to this? I mean, how could he actually say something like that? No, I mean, I've talked to many PhDs in biology, not this one's in the Great Debate community. And I've asked them all about this before. I've gotten other people that you guys have met before, talked to, that I know that just won't come on. But I've asked them about this kind of stuff, and they've all said the same things. And one of the things is that you wouldn't have repeats there. You wouldn't have long telomeric repeats because the fact that there was a degradation there, that's what allowed a high probability of a telomeric fusion. Because as you said,
have you ever? Sorry.
Go ahead.
I was just going to ask: have you ever considered the fact that he might actually know he's full of shit?
Yes.
Does he believe his own bullshit? I don't know. I think once he figures out that he's wrong, I think there's some panic response here.
Do you think any of these guys would ever go, you know what? we were wrong about this. Like I was just looking something up today where ICR did do a retraction. Are you familiar with what's his name? I was just looking up. Georgina Prudhomme uses some arguments when it comes to like the molecular clocks and mitochondrial DNA to show that supposedly that mitochondrial Eve only lived like 6,000 years ago, and she fails at it miserably. But some of the work she did on that came from a guy named Dr. Nathaniel Jennison or something. Yeah, that's it, Jason. And
he's terrible. He's terrible.
Yeah, and he came up with a hypothesis, and it had to do with the amount of frequency of mutations in species that reproduce very quickly, and they had actually acknowledged and said that hypothesis had been rejected. So they did admit that he made a mistake, had a hypothesis that was incorrect, and that's the only indication I've seen anything from them that they ever said that there's somebody in their club was wrong.
He wrote a massive paper a couple of years ago. It was so long and so long-winded, and I went through it over the course of a couple of weeks and eventually pointed out the one fundamental flaw that he made. He's pretty bad.
But they at least admitted that he was wrong, right?
Well, that's good.
The ICR even said yes, that hypothesis was invalid. I wonder what would happen if Tompkins actually, came out and said, you know what, flawed.
Yeah,
I don't think they would. But
if he did, I would just imagine
Harvard or something in a relevant field.
Harvard.
I'll look it up. Hold on. I'm pretty sure.
If he did find out that he was wrong, either he would say he was wrong, or he wouldn't say that he was wrong. But still, I think the same thing afterwards would happen. He just would ignore the fact that he was wrong and ignore what exactly telomeric fusion in humans and other animals suggests, and just try to find something else that supports his belief.
These people, I think what it is these people write these blogs for the layperson. Like I said, I've talked to this one person who reads these blogs, doesn't understand it, but just uses them as evidence. And that's the target audience for these younger creationists, right?
Nathaniel Jameson
people that don't understand what they're saying, but it looks impressive.
He's got a PhD in cell and developmental biology from Harvard.
Yeah, so he's not uneducated.
No,
right?
But he made a very, very poor mistake.
Maybe it's not a mistake. Maybe it's a calculated error.
Well, that's what I'm saying. I think so too. That a lot of these young creationists just they don't care at this point. They want young creationists to be true so badly that they're willing to throw away the objectiveness that they have. They're willing to throw away
their career.
Much of a career,
Well, being a young creationist and having a career in biology or physics or anything doesn't exactly work.
Well, they're apologists. That's what they've degraded it into, is just being apologetics, and they have to maintain that.
Book sales way outweigh what the book sales of a great young Earth creationist is way higher than that of a crappy scientist. So if that's where you're at in your career,
yeah.
But it's also a lazy career too. If you're a creationist scientist, I don't see them spending lots and lots of money doing lots and lots of work to produce viable results that actually lead to technologies and stuff like that.
How much do armchair scientists get paid?
Zero.
You look at Danny Faulkner, a doctor in astrology. Like, he's been hired by Answers in Genesis forever. He will.
Wait, they hired an astrologer?
Yeah.
Astrology.
Oh, astronomy. Yeah. Sorry.
I was like, wait a minute. You get a doctorate in astrology now?
No, astronomer.
Okay.
You can't get a doctorate in astrology. They'll just look at you and laugh.
They have a couple doctors of astrology.
But the point is, he's got a PhD, and that's all they care about. They trot him out so that they can have a PhD on stage.
Right.
So he has a PhD.
But it's in a related field that makes people in the audience feel like it's all good.
When they write these blogs and they have that little name, here's this PhD that wrote this. Most people that are in the field can read this and go, "Oh man, this is crap." But they're not writing to those types of people, right? They're writing to the people, the average Joe Schmuckatelli, that just says, I believe young Earth creationism is right. Let me go read this. Oh, I have a PhD saying this, therefore, ergo, it must be the case. And
But
it
then we
provides
have the
some kind
rest of the
evidence.
PhDs saying something else.
But they don't care about that, though, Max. They don't care. All they care about is they have a younger creationist saying this.
And even better,
Wayne!
there's a guy I'm actually doing a video of tonight. I'm refuting, what's his name, Dr. Robert Carter, who has managed to, in the mitochondrial DNA field, he actually has one secular paper. He will never have to work again. He will be paid for by Ken Ham forever now, because he has a secular published paper, one peer-reviewed paper, and it's actually in the computer science of DNA. It's not even in the biology of DNA, but it doesn't matter. Like, he's set.
Wow.
Because DNA is a language system. Ah!
Well, I actually read his
Sorry,
paper.
these people sold their soul, I think.
With computer science for storing the stuff and for recombining sequences in the correct order. Great, but that doesn't mean he understands the biology necessarily.
No, they just they make up these laws of information theory and then say that this is correct, but I can't find anything about like laws of information theory outside of creations. This stuff.
Well, no, there's informational laws.
Well, I'm sure there are, but I mean, like,
that.
the most popular searches on Google are from things like Answers in Genesis for the laws of information theory, and I don't think a scientific law would ever say anything like it can only come from a mind. It sounds a bit contrived.
I'm not a fan of Microsoft, but the least favorite thing Bill Gates ever did was accidentally say on a show that DNA is exactly like computer code. And they trot that quote out constantly. Well, Bill Gates must know. It's like, no.
Yeah, because he's obviously a cellular biologist, right?
Mm-hmm. Bill Gates knows everything about that because he's got a lot of money. And if you have a lot of money, then whatever you say is true.
Well, I've noticed that again with the younger creationists. They're almost always, their argument is very similar to what we were saying before about trotting out a PhD. They love argument from authority. They love it, right? They never provide an argument for themselves, but they're like, well, such and such says, or here, let me show you this quote here. I don't even like to have those types of discussions anymore. I'm like, I don't care what this person says. I don't give a shit what what Dawkins said or Cohn or anybody, right? If I'm having a discussion with you, I'm having a discussion with you. So don't make an argument from somebody else is using. I don't care about that.
I know, it's ridiculous. I asked my dad for some shit, and he wants to show me some 1 hour flat earther video. I'm like, no, I was asking you.
Yeah, because I'm not going to sit through a 1 hour flat earther video when it's not going to tell me anything, right? It's just going to be bullshit.
It'll just tell me the fucking shit I already know. Yeah,
I think that's the thing we need to start demanding from them, is because like a lot of stuff on the scientific side leads right away to functionality. Like when we learn how to make new chemical compounds, well, those start getting applied as soon as possible to whatever best functionality. Like if you want to create a new way of creating polymer chains that is stronger, well, then people are going to take that to make stronger fibers, which can be used to make stronger ropes, or even materials that are impact resistant. Or that's why I said I think we might need to start pushing that angle more, because that's the one thing that side doesn't produce.
You're exactly right.
Asking. Yeah, and they don't produce anything functional. They don't produce new medicines. They don't produce new technologies. They don't produce new materials. They don't even show. They don't even show how your child is related to you.
They just say that, no, it can't be, because DNA doesn't show that we're related to everything else.
Precisely.
Yeah, I don't understand the argument on common descent either. I was engaged in that with somebody just recently too. They're like, well, yeah, accept speciation, and that species do, come from other species, but I don't accept common ancestry. Well, that is
what,
like, showing what common ancestry there is. And I mean, if you go back billions of years, what arbitrary thing are you saying did this process stop? I mean, if species come from other species, where do you draw the line and say, hey, you know what? No. Hey, we can't go back more than 6,000, 7,000 years of speciation. Where does this happen? I mean, I don't get that.
Well, I don't get it when you said that they accept common descent, but they don't.
And once again, people
they accept it enough to they accept it just enough to get it past any obvious proof you can provide. Like just enough that you're related within your own kind. But you see, even as I said, here's another point. Whereas what I was saying can be applied, because even common descent is highly functional. It gives us a way to track down things through species, whether it be trying to find a related virus so that we can find a way to battle one that is actually impacting us with one that won't, or say you're looking for a compound that only nature can produce, and you have a species of, let's say, a plant that has gone extinct. With the theory of common descent, you can use that to try and figure out, okay, what is going to be the most likely related species that might have the same chemical producing traits, or who can be coaxed into producing the same chemicals for say a medicine or something. See, that's usefulness. That is actually actual functionality. That saves lives or produces new materials. And I mean, that's what I say to the people that try and convince me of the physicians. I'm like, I'm sorry, I'm going to go with the technology. Technology is telling me that even if they're not 100% accurate, they're on the right path.
And the more accurate they become, the better technology will be.
Bingo.
That's kind of what I was thinking. I mean, creationists and the like produce absolutely nothing, and then they don't realize it. They just sit on their asses or in their armchairs and make up a bunch of shit about science and then try to tell everyone, and then they know they're lying about it, while there are other real scientists out there doing work that, like Mary Schweitzer, but then they just choose to misrepresent them.
Well, I can't think of anything they've ever added. Now, people will trot out, the creator of the MRI. Well, so what? He didn't win the Nobel for it. 2 other people did. And, he never used anything relating to young Earth creationism to actually.
Yeah, and he never said
God. He wasn't that active in the community. He just, one of those people that just thought that Earth was young, but, so whatever.
Yeah, so what?
Yeah, it wasn't his theory. It wasn't his theory about a young Earth that created the MRI.
Correct.
It was his knowledge of the MRI. Yeah. It was
statistically, you are going to have some people that have some fucked up beliefs and that can do amazing things and discover things. It does happen, but it's not the norm.
Nope.
Like, I don't mind challenges to stuff. I mean, there's nothing wrong with challenging stuff to make sure things are true or not. But it's when you have this obsessive level where it just cannot be true. And you must accept my way. That's the thing. That's that second part. That's what makes them annoying. It's like, you must accept my view because I say so. It's not that okay, I'm going to just continue down this. Like I think I made it known that I'm a bit of an anti-timist, but I'm not going to tell anyone that they have to be the same position as me. I mean, well, I'll go on to it another time, but I'm saying I don't demand it of anyone, and that's the thing. But they demand it. They say, I can't believe the Earth is more than 10,000 years old. You must believe it too,
And then you ask them for evidence of it. That's the biggest thing when I'm talking to people on these threads, that I'm asking for the evidence, and yet they're just not providing any. And then they'd ask you to say, well, you have no evidence for evolution. You have no evidence for the age of the Earth being old. And it's like, yes, we do. Overwhelming, massive. It's gargantuan.
But Darwin said this theory would be falsified if things were so complex.
And again, I mean, I'm so tired of these misquotes and people quoting things out of context and saying, well, such and such said this. I don't care. I don't care if Darwin's was a racist, homophobic piece of shit. He wasn't, but I mean, hypothetically speaking, it doesn't matter. It has nothing to do with his theory. He could have recanted on his deathbed and said, hey, he was completely fucked up. He could have said that, I believe in Jesus and evolution's wrong. It wouldn't matter an iota to the actual fact that evolutionary theory is a valid model that explains evolution, which is a fact that occurs. Nothing will change that from what Darwin thought or thinks or whatever. Well,
Brahe at the end of his life became a geocentrist, but that doesn't mean we should accept geocentrism.
But anyways, hey, Hog, did you drop in?
But I took a nice nap today. That was quite pleasant. I was actually keeping an eye on the feed today, and you mentioned was it yesterday that Roohif was going to come talk about the experimental design and what he hopes to find, and I think I slept through that part, and that's a shame.
Well, you're gonna have to watch it. It's first. Well, I don't know. We talked about a half hour, at least. The first part's worth watching because he does go into detail about. He can do a quick recap if you want.
Hog tie. Do I hear Jake West in the background?
That would be unfair. Jake West? I don't get that. What? My place is kind of noisy. I'm going to be pretty injudicious with my own mute button because my place is a little noisy right now.
I thought I heard Jade in the background. That's all.
No. Oh, Jade West. Oh, okay. Sorry. I think you said Jake West. I didn't know
Jade
what that was.
West.
Jade West. We're some distance, yeah. No. Okay. So what was I thinking? I certainly want to know more about the methodology. So I'll go back and watch the first half hour. But I know when I tuned in the why question, the sort of what do we hope to learn here? And it reminded me that and people might remember this, and it's funny because I was trying to remember who was it. I was in a hangout early in the days of this whole chromosome 2 fusion thing discussion. It was quite a while ago, and at one point, of course, they were throwing up this argument that well, you just don't see telomere fusions, right? There are some examples of telomere satellite fusions, and but it's just something you don't see. Well, of course, then they claim that you see it in cancer cells, and that's such a misguided bit of thinking. But which let me not derail myself onto that. But at one point, in arguing with him about this, how you just don't see this. You just don't ever see telomere fusions. I said I made the point. So just as an aside, okay, what if it is a telomere fusion, or so we see this, and what if it's the only one, or I think I said, well, what if it's the first one that we detect, right? And what I'm saying there is you can't strike out, you can't absolutely rule out some scientific discovery simply because it hasn't been seen before. And when I asked the question, well, what if this is the first one we find, right? Their response was literally, there were no words. It was literally laughing, like guffawing and knee slapping. And I'm trying to remember who was in the room for it. I think Ronnie was there. For some reason, I think Beckel was there, but I may be misremembering.
I think it was Antonio's. Was it Antonio's hangout?
Yeah, it was. It was on Antonio's channel. That sounds right. I wonder who else was there. Darwin's deity might have been in there, but it was just like, what a ludicrous thing to say. And I sort of thought, well, that's funny because I understand if you think the other person's reaching when they're making a point, that it is kind of funny, but then it sort of revealed to me how little they know about the state of the science, because we haven't, now that we've got the ability to sequence DNA, we didn't then immediately sequence all the DNA on the planet. Right?
There's a lot.
There's a lot.
Yeah. Well, there's been a lot done, but in terms of what we know, there's something like 5 million extant species. Right. We haven't mapped all these things out. We don't know.
That was 8.7 or something. Yeah, that's
is it, well, yeah. It's in the low. Well, let's put it in that order of magnitude, right? In the single-digit millions, or maybe up to 10 million. But we didn't run around and sequence everything, and try to make a big map of oh, well, this is related to that. Based on strictly on sequencing. I think that there'd be no surprise if it doesn't. When you do that, if you can do that, then it's going to match nicely with the tree of life presented by other methods like anatomy. But anyway, I just thought it was funny that there was this thinking that well, you just never see these things. You never, ever see these things. Okay, well, what if the first one we discover is with humans? And then what if we look around and we find out this is actually a really common phenomenon? Or what if it turns out not to be a terribly common phenomenon, this telomere fusions, but it does tend to happen in mammals, right? It may be strictly a mammalian feature. And then we can say, well, why would that be the case? Why don't you see this in in birds or crocodiles or whatever else? Because it makes us.
That would be an interesting question. That would be an interesting question. Wouldn't it be though? Why do we see this in mammals and other things? Don't you think?
Well, yeah. Well, I've asked the question of CRISPR and rational mind about. I may have asked Fiona as well. Yeah, Fiona was there in this thread when I discussed it. Is it, well, what is it? What's going on to make a telomere fusion? Because the telomeres are there to prevent such chromosomal fusions from occurring. And I was making the point that is it possible that because you know that when there's mutations to any to the coding, something that the DNA that codes for any particular protein, you might get a slight difference in the protein that might make it more effective in its purpose or less effective in its purpose. And sometimes proteins have more than one purpose. So sometimes there's a bit of tweaking that goes on in the protein structure. Just one second. He left.
Oh, sorry. I was going to say there are examples in other species outside of mammals. I know there's one in the jack jumper ant, which I'm, aiming for project number 2 to sequence.
Actually, now that Hogtie's been talking about this, I've been kind of wondering. It's been in the back of my mind, but now I may ask it. Is there a way to detect a split?
I don't know.
Split.
I mean, you could infer one. I would imagine you'd infer it by seeing the 2 chromosomes split, but without any fusion signature in the one that remains a single chromosome.
I'm not even sure what a split would look like. And I apologize. I wanted to sort of quiet down some of the noise that was in my house there, so I sort of interrupted myself mid-point there. But I think a split would be pretty rare because the overall anatomy of if I can use that word, the overall anatomy of a chromosome is more complicated than just like if you're thinking that everything's stored like on a reel-to-reel tape, and you cut it into pieces. Well, it doesn't matter how many pieces you cut that tape into, all the information is still going to be there. It's just assembled in different ways. You could cut it into 100 pieces and then splice pieces number 48 and 49 and whatever. All the information collectively is still in there. But chromosomes have their own anatomy. They have a centromere. They have the telomere segments that are quite long. So I don't know if you were to take a chromosome that had a single centromere and the telomere ends, then what would it look like when you split it?
Probably a weird looking centromere and one telomere.
Well, you wouldn't have any centromeric sequences.
Yeah.
Right, you have no place for
I wouldn't mind researching that, actually. Like, I know there are plenty of comparative karyotyping papers where the 2N diploid number is quite high, so up in the 80s and 90s. I should read those to figure out, or to at least hear what the authors have to say on how it got so high. I would imagine there'd be either a whole genome duplication going on, or a fission of a single chromosome a few times. But yeah, it'd be an interesting research topic.
Thanks for answering that, because I was always curious. So.
No. Well, sorry if I can make my earlier rambling point about the protein. So you've got, anytime you're coding for a protein, the protein has a certain job, and the efficacy can change. Like when there's a mutation in the DNA that codes for it, then it might be more effective or less effective. So I'd ask the question, and I don't know that I got a real solid answer from those folks yet, but that is it the case that I've lost it now. I had a nap and there's so much noise in my head, it's distracting. What is the protein that rebuilds? I want to say telomerase. We kept arguing about the pronunciation.
Telomerase too?
Yeah, telomerase.
Telomerase we're going with? Okay. The last 4 hangouts we discussed this and everyone argued over pronunciation. But it's the thing, so apparently these telomeres, as part of the chromosomal replication, there's always a loss at the end. Right, the telomeres essentially act like there's thousands upon thousands of repeats of these the telomeric hexamer, because they end up being like handles that get trimmed off during replication, and then the telomerase rebuilds up the telomere so that you always have a certain amount. Well, then the next time it replicates, it loses a bit, and then, well, if you had a less than perfectly efficacious telomerase, you might have a set of creatures or an individual creature that has more of a struggle keeping up with its telomere manufacturing.
Yep.
And eventually the replications, it would kind of get ahead of itself with losing these shortening of the telomeric handles, basically. And that would be fine. It just would happen to be that when you've got the 2 chromosomes that have shortened ineffective telomeres, on occasion, one of them would bump into another in just the right way, and then now you've got a telomere-telomere fusion. and it would be still a pretty rare event. But I'm saying that because it has to do with an allele, that codes for a weaker telomerase, that allele might only appear in a from a certain branch of the tree, within a certain clade. And I don't know if is it only mammals or, well, as Roohif points out, it includes ants as well. Well, if it's that broad, if it's possible that when you've got a species that's a mammal and a species that's an insect, Hymenoptera, that they're that far apart on the tree, I would expect to actually see plenty of creatures that are capable of having telomere fusions, if it's the case that that's caused by a weakening telomerase protein.
Well, there are diseases that have that, isn't there? That for some reason doesn't produce the telomerase as much as it should.
You can like experimentally knock it out and disable it. That will obviously, hasten the degradation of telomeres. But I mean, I'm sure it's the case that you could have a mutation of a single amino acid, which would obviously degrade the performance of it, like Oktay said. As to how it actually happened in a human, could be that they just broke off where they broke off. Who knows?
But do you remember what Tompkins actually says that he actually? Well, okay, if you actually look at the actual sequence of the fusion reason on human chromosome 2, what is the explanation of why it looks the way it does? That's something I just can't seem to find from him.
Well, he seems to think that it was what we see now was actually a pristine sequence before, after the split, and that's just not the case. I mean, the degradation that we see, the imperfection, like the non-pristine sequence, was not pristine at the time of the fusion. The sequence we see now is subtelomeric, so they're not perfect pristine repeats, and that sort of subtelomeric degraded sequence you can find on the ends of current chromosomes before you get into the pristine repetitive telomeric repeats. So it even adds more evidence, I think, to the fact that it's a fusion. The fact that you find these subtelomeric degenerate arrays on other chromosomes. Does that make sense?
Yep.
Cool. I'm going to go and have some lunch, actually.
Lunch?
I'm pretty hungry. Lunch. It is 1:24 p.m.
Oh,
in Sydney.
Yeah, that's right. You're in Australia.
Yeah.
Me, it's 9:24.
Hungry.
Yeah.
Guys,
thank you, my friend. I'm sure people got something out of this. I'll keep it open for a few more minutes. This was interesting, and I really look forward to this kind of project that you're doing. As a matter of fact, this is like cool that we're starting from scratch. You're saying this is exactly what I'm doing. I'm getting the materials for it. I'm going to document my work, and hopefully might even send it off to Independent Lab to have it validated. I look forward to Tompkins, looking at this after you're all done with it, and I also look forward to Tompkins' paper that he's putting out next month, supposedly to refute some things that you already addressed. I'm really looking forward to seeing what he says, because he got real defensive real quickly on that.
He has yet to have a substantive response to anything that I've written. So
I know I've seen his responses to you. There's nothing substantive in anything he's written to you. It's all been mostly ad hom.
Well, if you're wrong,
I'm not
then
expecting
you're just
much.
going to keep being wrong.
Yeah.
LOL.
Pretty much.
Yeah, it's kind of depressing in a way, because I was like, come on, Tompkins, I mean, I start off really civilly. I'm still civil to him, but I'm like, Roohif makes some interesting points. Other people in our community has actually said the same exact thing. So, his level of education should make no difference to what he's actually saying, when other people who do have PhDs are agreeing with him.
Yeah.
So
just respond to the points. Yeah.
Yeah, it'd be even more hilarious if you actually ended up, even if it was a decently prestigious journal, got published.
Yeah, that'd be funny.
Something interesting. Yeah, that would be like grinding salt in his wound.
Yeah, I mean, I don't plan on.
Yeah.
I don't plan on attacking the creationist position in my paper. I'm just going to say, look, this is here's some extra DNA for the pig genome, and look, here's the sequence of the fusion site if I find it. That's pretty much going to be it.
Just be professional, and then wave it in their faces after the fact.
Well, he's always been. I can tell you right now, he's never been negative toward Anderson or Tompkins. And I mean, shit, Anderson started off like hostile to you. Well, not in anything I've seen as far as your emails to him.
See, that's the best.
You haven't called him any names or anything.
I've said plenty of times that I believe he's dishonest in his work.
Oh, now, but I'm saying in your correspondence with him, though, what I'm talking about.
Oh, I'm civil, yeah.
Yeah.
Anyways, that's the best way to be nasty is when you're absolutely polite, absolutely professional, and all you do is just lay out the evidence.
Yeah,
basically. And not have to do much defending because it's there. That is the best way to be nasty, because when they have to start screaming and making all sorts of cries and accusations, and it starts to show itself off.
Yeah.
The fact that he's only come back so far is that you don't have a degree in biology. I mean, that says a lot. So.
Yeah. Well, if he's doing good work, he's doing good work.
Exactly. All right, I'm going to bail because I'm quite hungry.
I'm going to bail too, because even though
All right,
yes,
thank you, sir.
Oh yes, Roohif, eat well.
Oh yes, thank you. All right, see you guys.
Well, thank you, buddy. It's so amazing though that we have a lot of people in the community that are willing to actually address the actual science with Tompkins and other younger creationists, and yet every time they come in or around, none of them want to talk about the actual science or the evidence that they have. It's very rare. It's always been more, well, somebody else says this, or it's theologically speaking, the Bible says this or whatever, but they don't actually say, okay, well, here's actually evidence that supports a young Earth. It's always been, let me show you the evidence why the Earth isn't old, which was bullshit, but I don't see any positive evidence for their own position, rather than just promoting what was called negative evidence against deep time.
Yeah, that doesn't work. Just because you falsify one position doesn't automatically confirm yours. But I have to go because my phone is dying, even though it's plugged in. So I gotta go kill myself. I'll be back.
Well, nice meeting you.
Well, I'm probably gonna keep this short. I just wanted to have a quick video on Riff's thing. And then obviously with C. Brown continuing to make accusations he can't support. And I think he's just in full troll mode anymore. I mean, now he's just basically saying, well, yeah, Fiona's intimidated by my level of knowledge. And it's just like, okay, at this point, we know he's trolling. We know he's full of shit. I mean, C. Brown is a dumbass.
I'm not even afraid of C. Brown.
So, yeah, I mean, it's just comical. And I explained to him, Lude, you're comical. I keep you around for entertainment purposes, but nobody's ever going to take C. Brown seriously. It's just not going to happen.
It's funny that he thinks somebody might.
I don't know if he really does at this point. I mean, when you start saying the things like, "Oh, well, yeah, Fiona's intimidated by my level of knowledge," and you barely can have, a GED. I don't think he even had his GED, did he? But I mean, there was nothing about these topics. Nothing. That's why half the shit that he says, we sit there for a half hour trying to figure out what the fuck he's talking about. And it's not just the fact that he's talking to his phone and his phone is doing a shitty job with a voice translator, text or speech to text. It's the fact that he's just using these terms so strangely that even I'm like, I have no idea what's going on. Oh.
Well, I'm going to go.
All right, boys and girls, I think I'm going to bring this to a close. Anybody got any last things? Paul, Gia, you got anything you want to plug real quick?
Yeah. No, I got to record. It's funny. I'm actually recording the new video on mitochondrial Eve. So when you guys started talking about that, I figured I better jump on to make sure I got my ducks in a row.
I think I got something from you. I'm gonna read it tonight.
Cool.
I'm not that hip on that particular thing. I know. Again, we had touched on Dr. Perdome is talking about. She thinks that the mitochondrial clock, the molecular clock of mitochondrial DNA, indicates that mitochondrial Eve only lived a few thousand years ago, right?
Well, what's funny though is they tear apart that clock, the molecular clock, in every other circumstance. But when it works for DNA, they're fine. Like it's just, I actually have clips of her saying both those things, so it's ridiculous.
Well, that's hilarious. Cherry picking.
I mean, I don't know her full argument. I just know that what she's basing upon, that has already been acknowledged by the ICR to have been wrong. She's with the AIG, and I can tell you right now, AIG is even less responsible in their information than ICR. ICR is bad. I mean, really bad. But AIG is illegal itself. They don't give a shit whatsoever. I mean, they really don't. They are horrific in the things that they put out, and it's just Ken Ham is an even bigger con man than anything that ICR puts out.
Yeah, but what's funny, he gets government money instead, so that's, you know.
Yeah, he has financial incentive to be dishonest.
That's right.
Yeah.
All right. And I think, I don't know, I'll probably be on Sidestrike's channel later.
Well, Friday or tonight?
Isn't this Friday?
He usually does. His debunking on Fridays. Yeah.
Yeah, it's Friday.
Yeah, it's Friday today.
But it's Saturday.
Yeah.
No, it's Friday, dude.
No, it's Friday.
Oh my God, I'm a whole day off here. Fuck my life, really.
Right. I know what you're saying. I know that feeling. I experienced that.
You're right. It's looking good. Well, okay. Oh, strange.
A couple times I woke up and said, I thought it was November. So I know that feeling completely.
All right, let me check my live feed real quick, see if there's anything interesting going on. Blah, blah, blah. Let's see here.
Thanks.
Does anybody follow that thread with C. Brown just real quick? I mean, anybody know what we were talking about? No.
I posted a few rebuttals to him, and he just ignored me because he doesn't know who I am. So.
No. Paul, when is your next video coming out? You said.
I'm gonna record tonight, and we'll see if it comes up Monday, maybe.
Okay. All right. Anyways, I know we have a lot of people watching, but I have a friend that wants to go do some stuff. So, anyways, guys, thanks for joining us. I hope this was entertaining. I think Roohif did a really amazing job of explaining what he's going to be doing. So, if you got any questions on that, just send it to me or send it to him. I know that he's different time zones, so I'm up a lot more often, so I can get him at all hours of the night. So, anyways.
We're going to do that? Yeah, sorry. We had an incident here. I had to step away. I was still listening off, but I wanted to say one quick thing about what do these guys do with their day? You guys, as you were talking about, once somebody gets a publication, they go work for ICR, AIG, and they're kind of set for life. What's interesting about that is when you look at what scientists are paid to do, they're paid to do more research and write more papers and teach classes. And go to research meetings and go to administrative meetings, contribute to the whole culture of the community, teach graduate students, and present their material to the public. Now, that doesn't mean they have to do every interview when somebody calls up, but they do see that as an important branch, communicating, disseminating the information from their research to the public. And what I cannot figure out is what is a day like, a day in the life of a quote unquote scientist at AIG or ICR, because
no idea. I don't know what their pet products are.
Because what happens when you invite them to say, well, come on, let's talk about, you've written 2 papers in the last 3 years, and we'd love to talk about them.
Yeah, if you look at their
blog posts, yeah. Sorry, Paul.
If you look at their schedule, they are literally booked almost every weekend. Like, I actually follow Georgia and some of those, and like they're traveling to churches and to various places almost every weekend. I think on a Wednesday.
I don't disagree with that.
Like, I think on a Wednesday.
They don't do any science.
But they've also started a news show every second day, so they're doing that too, which is hilarious.
Oh, so here's the point. Yeah, pimping the message. Good for them.
Yeah, they do get out there. I do agree, and I do see them at churches, and I watch some of their videos, and so yeah, I know that they are doing emissary type work for ICR and stuff like that. I get that. But here's the thing, they know that this was a big thing with the Great Debate community. I mean, Tompkins is well aware of who we are. He knows who Roohif is very, very well. I would think that this is something that they should really look into more, because this is directly going to their claims, right? I mean, if they could show Roohif and a lot of the members of the Great Debate to be wrong, this is a feather in their cap. And so I don't know why they would pass up that opportunity.
It's not even about showing people to be wrong. It's about being willing to go out and discuss your ideas. And there are some forms that you don't want to go into, but for the most part, you do want to take up invitations for conversation. And I mean, I remember corresponding with Tompkins for a hangout on my channel, invited him to do an episode of the Magic Sandwich Show, and initially. And then you end up hearing from do we say her name? Beth, right? Yeah, I'm gonna say her name. I'll give at least a first name.
Beth, yeah.
And yeah, I don't know if you mentioned it in this hangout or not, but yeah, they just have a person there whose main job seems to be, no, we're not talking to you.
They're hogtie public.
Well, it's funny though, but wait, what's like? It's funny because the other place that not I think it was Beth's from ICR, right? Yeah, from the other one from IC Answers in Genesis. I can't remember the person's name, but they gave me a reference to the person who actually the big wig that does all this stuff, and he had actually corresponded with me, and he said, "Well, tell me more." And he said, hey, can you show me the video from Dr. Schweitzer?" And I sent him that video, and then I get about 2 hours later, I get a response from somebody else saying, we can't do this, we can't accommodate you at this time, but they were interested. They did watch the video of Dr. Schweitzer's interview, but I think what came down to being is they're not going to go on programs where the host or the venue is not a young earth creationist mindset, right? They'll go on like Ian Juby and those kind of places, but they'll never set foot in this kind of forum. And I think that is very telling, because I think you were right, Togtai. The thing is about just having the communication, right? And I do believe that would be a fused feather in their cap. But it's still about just, having that communication. That's the conversations that they don't want, though. It is very one-sided conversation with these people. They don't want to have a dialogue. Have you noticed that at all?
Yeah, and I don't have any objection to them choosing their forums, right? Like I
don't either.
Because yeah, that would be conventional for any academic. Again, when I don't mean to say that I think they're academics or scientists in any way. I'm saying that they're trying to follow that model, or I'm saying at least we could try to hold them to that standard. It'd be unfair for us to say, well, that's ridiculous that they don't just like jump into every Google Hangout they're invited to, because you're not going to find professional scientists that will do that. That's a tremendous waste of their time. Plus, it also sets up the potential for the dissemination of actual scientific information to be misrepresented, right? I mean, everybody knows that clip of when Richard Dawkins let that film crew into his house, and then it turns out they dishonestly presented themselves, and he realized partway through, well, these are creationists, and that sort of bothered him. So he wanted to stop the interview, thinking, well, they tend to do pretty sketchy things, and how do they respond to that? They did a pretty sketchy thing and took that bit of the interview and cut it up to make it look like he didn't know the answer to a basic question he was being asked.
I remember that. Yes.
Yeah. So and he's come out in interviews since then, and he did a presentation. I can't remember which conference it was, where he talked about why you can't give interviews to people like that, right? And I think that's true. You simply can't take everything that comes along, as I said, just because of the time management thing, but additionally because of there are some forums where people are there to misrepresent you, to dishonestly represent what you're trying to present from your particular field. So if we were to say, well, that's not right, that for example, Tompkins doesn't just join every Google Hangout. Well, wait a second. We don't expect Professor Moriarty to join every Hangout, or you know what I mean? Like we wouldn't expect it of other people.
I completely agree with that. However, the difference here is that again, he is well familiar with Roohif. This is not like just some hangout stuff that some arbitrary people going, hey, can you come in and talk to us? This is something that has been a thorn in his side. Roohif is a thorn in his side. He's not fond of Roohif. So I think it would be very interesting for them to have an actual conversation, a two-way conversation. And he was willing to do so. He actually said that he would a couple weeks ago.
You need to catch someone to have a new book coming out, Steve.
Wait, 1 sec. He actually said that he would. It wasn't his decision not to have the conversation. It was ICR.
Yeah,
right?
But these guys do book tours. They go on every show when they have a new book coming out. You need to watch when they have a new book. And these guys publish or perish just like anyone else. But they're selling to Christians. But they put out like 3 or 4 books a year, each of these guys.
I don't think we have to hold them to, getting into every hangout. But what we can do is we can actually measure the quality of the so-called discussions. We can point out that this so-called interview you did was just all one way. And I think that's probably a better approach. At least that's just my idea, is to hold them to the quality, to the style that they go for. And I think that
it is telling that old Earth creationists are a lot more willing to come in and talk about their their beliefs and the science than young Earth creationists. I mean, we've had a numerous amount of old Earth creationists come in, so I found that to be interesting.
Yeah. No,
But with that, I think that's going to come to a close, people. Oh, I forgot. Let me check the.
Yeah, sorry for keeping you in here longer, but I really enjoyed.
Oh, no worries, man.
This was fun.
Yeah, no, this was good. That's why I kind of want to keep it short because I like these smaller ones that people can kind of listen to for an hour or two while they do other things. I get more positive feedback from those type than the whole long 6 to 8 hour ones that we have, right?
Steve, you may want
to
advertise the debate between Floyd and myself.
Oh, I forgot about that. Yes. What is the date on that again? That's going to be February what?
24th, I think. We
24th. So that's Friday. 3 weeks from today. Yeah. So go ahead. And it's 3 weeks from the day, right?
Yeah.
Yeah. Go ahead and plug it real quick.
You choose the time. I don't mind.
It's all good. We'll figure it out. What it's the 24th, so plug what you guys are actually debating, because it's going to be more of a much more structured debate. I'm just going to be more of a introduction, timekeeper, and closer. So I don't have anything to say about this topic whatsoever.
So apparently that was the quickest, shortest agreement on the format I've ever seen. But
I noticed that he said, "Hey, how about this structure?" And you said, "Fine." I was like, "Wow, that's pretty cool." Yeah.
But yeah, we try and stick it to 20-minute, like a lecture format each, depending on. And I've given Floyd freedom to go to maybe 30 minutes if he needs it.
Then what's the actual debatable topic that you guys are going to be?
The topic is on Noah's flood. So I'll be defending the local, regional flooding, and based on recent research, I'll be showcasing evidence for it at the end of the last ice age. So
now let's keep in mind that Floyd's position is that the Bible speaks of a global flood, not that he actually advocates that there was a global flood. Correct?
Yeah. So I think what Floyd,
I want that to make sure we understand that that's going to be the debate is not okay, because Floyd is not a person who thinks that there actually was a global flood.
I think Floyd's going to try and showcase that the Bible, or I don't know, I'm just going to zone in on the Bible here, but he's going to try and showcase that the Bible teaches a global flood. He may appeal to outside the Bible, I'm not sure. But based on that, I guess also that it's not historical. So it's a myth that advocates a global flooding. I will be
very great to listen to. I really do. I think this is going to be an awesome actual debate. I mean, we are part of the great debate community, so some people do debate, but I want to note on this. This is something that people are debating that is debatable, right? Does the Bible say this or not? When I get shit from people, like I got shit from Trad Owl for not debating kind of thing, and I try to explain to him what I consider to be things that are debatable and things that are not. Flat Earth, not debatable. Young Earth, not debatable. These are not debatable concepts. They're falsifiable, which I believe that they are, and I believe that people can talk about them, discuss it, and if you want to give evidence for young Earth, fine. But you cannot have a debate whether the Earth is flat or not. You cannot have a debate whether the Earth is young or not. These debates have long been over. Okay, so I use the term debate very specifically. Other people do not have to use it the same way I do. I get that, right? But when I mean a debate, I mean a formal, structured things like you guys are doing. Now, no matter who wins this debate, it doesn't mean that it was a local flood. It doesn't mean there's a global flood. Doesn't mean there was no flood. Doesn't mean there was both. It just means that who's presenting a better argument to substantiate what they are saying or what they believe, right? And so I want to reiterate, it is not about something that's saying, that's already been confirmed, right? We're not debating whether the flood did not happen. I believe that there's an overwhelming evidence that the flood did not happen. I think that's pretty well substantiated. And confirmed that there's no possible world flood, but they're not debating that side of the argument. They're saying which one does the Bible claim is true. Is that correct, General?
Yeah. I want to model.
Okay.
I want to, for the sake of other Christians out there watching, I want to model how a Christian should debate, because I've been noticing a lot of Christians, they don't debate. They debate just to they want to win. When a Christian debates, especially when you see the early church, how they debated things, it was all for the sake of understanding the other person and, you know, going along.
It was to get to the truth.
Yeah, it's not about, oh, look at me, I won the debate. Model that. Look, Floyd, I would consider Floyd to be a really good, informed critic of the faith and how to model interaction with someone like Floyd. And I think in that way, and you've seen me talk about this all the time, that, you know, the building of bridges. So I hope that this could be like an example of that.
Yeah, I totally think that, you know, this is going to show people what two rational, intelligent people that do have different views can have a debate, and it'd be interesting. You respect Floyd, Floyd respects you. That's the kind of conversations that I think that most people in the great debate community would much be more willing to listen to than people having a debate where, oh, let's debate whether the earth is flat. Let's debate whether they're, you know, creation is true or not. That's just boring shit, because again, I don't think those are debatable topics. But all right, so that's going to be on the 24th. I'm still working on Jen Peoples. Paula is still listening. Maybe he can get a hold of her, because I don't want to bug her too much. I'll check her out, or I'll check her out. I'll check into seeing maybe on Monday, sending off a message to her again, because like I said, I don't want to like be too persistent, but I think it'd be fun to get her on. All right, guys, thank you for joining me, and I will see you guys later on. See you probably Sunday. Bye.